Welcome viewers. Today we will be discussing on the topic study of model organisms to understand the aspects of life sciences. As we all know that model organisms can be divided into different categories like bacteriophages or viruses, bacteria, protists, fungus, plants and animals. Today we will be discussing plants as model organisms. As we all know that plants have got versatile ways of surviving in versatile places. They can grow. They have got tolerance to survive under various conditions. Means they have got many ways to adapt with the changing environment. So obviously there must be something that helps them to survive under such diverse situations. So to understand that, we must first try to study them as models. So before going into the topic, let's classify them as the plant kingdom can be divided broadly into cryptogamia and phanerogamia, means the non-flowering plants and the flowering plants. This cryptogamia means the non-flowering plants can be further divided into Thallophyta, Bryophyta and Pteridophyta. And Thallophyta is a big group wherein there are various forms of plants. They have got a thallus-like structure, means they don't have a defined size or shape. Then in this group we will be specifically focusing on red algae, brown algae and green algae. On the other hand, the phanerogamia group means the flowering plants can be divided into Gymnosperms and Angiosperms. These Angiosperms can further be divided as monocots and dicots. Gymnosperms, what do you understand by Gymnosperms? Means they have seeds which don't have any closure. Means these plants don't produce fruits. Their seeds are naked seeds. On the other hand, the Angiosperms, they produce seeds which are covered, means coated. If we consider the evolution, then Gymnosperms were more primitive than Angiosperms. And in Angiosperms, we have said that they can be divided into monocots, means they have got a single cotyledon, and dicot have got double cotyledons. And in the monocot can be further divided into two parts, means aquatic monocots and terrestrial monocots. Similarly, the dicots can also be divided into two parts, aquatic dicots and terrestrial dicots. So we will start our journey with Thallophytes. In this community, the first member is green algae. And the example of green algae is Acetobularia acetabulum. What we have got is the class Ulvophyceae, order Dasyclades and family Polyphycia, and the genus is Acetobularia. It is found in the subtropical waters. It is a single cell organism and it is gigantic in size. Next member of the Thallophyta is red algae. The member is Neurosiphonia harvey. It is widely used as a model organism in genetic, molecular and biological speciation studies. And the next Thallophyta member is brown algae. Its example is Laminaria digitata, and it belongs to the family Laminiaceae and genus Laminaria, and the species is Laminiara digitata. And what are the properties that make it a model plant? Is its early embryogenesis, its endosymbiosis and the origin of chloroplasts was first studied in Lamiria digitata. Its biotic and abiotic stress studies are also done using these organisms. It is also important in the production of various bioactive compounds. The second group, that is Bryophytes, and the first member of this community that we are going to discuss is Physkometrella patens, and it belongs to the family Furnariaceae and genus Physkometrella. And the important properties are It shares fundamental genetic and physiological processes with vascular plants, and it helps us in study of evolution. It is efficient in study of homologous recombination, as well as gene targeting is quite easy in this Physicometrella patents. Gene knockout studies are also done with this model organism. It helps us in regenerating filaments and can be directly assayed within 6 weeks of gene targeting. Means we get quick results of gene targeting. It is used in molecular farming as well. We are discussing about Pteridophytes. In this community, we have got Selagiana moeldorfi. It belongs to the order Selagianales and family Selaginacea and genus Selagiana. It is important to study the evolution of terrestrial plant, and it is an important model for understanding comparative genomics. Gymnosperms. In this community, the member that is most popular is Pinus roxburghi. It belongs to the class Pinopsida, order Pinels, family Pinacea, genus Pinus, and species Pinus roxburghi. They can grow well in much more productive environments. Means the genus displays a wide range of ecological adaptations. It can grow in various ecological niches, and various features of pines are there that make it easy to study for human system. The gene alterations and the fate of planting are generally very well documented. Means informations are known to us. That's why studying with this system is quite easier. It helps us to understand the success and failure of the experiment very nicely, as because we have got an enriched database, and silviculture is improving productivity of plantation. The Angiosperm community. Here Angiosperms, as I said, can be monocots as well as dicots. So we will talk about Angiosperm monocots, and the most important member is Oryza sativa, means the rice. Then it belongs to the class monocots, order Poales, family Poaceae and genus Oryza. Oryza sativa has got a large set of genetic, molecular and genomic resources, and taxonomically it is distinct monocot. It is a model for understanding the cereal biology. It is supposed to be the second best model organism after Arabidopsis thaliana. As we all know that Arabidopsis thaliana is supposed to be the most popularly used model plant for studies on plant system, and Affymetrix array are publicly available for Oryza sativa, which again makes it a most suitable candidate as a model organism. Now we are coming to Brachypodium distachyon. It belongs to the order Poales, family Poaceae and genus is Brachypodium and species Brachypodium distachyon. Why it is chosen as a model plant? Means its genetic, cellular and molecular biology of temperate grasses, as it is used to study. It is used to study the genetic mapping and sequencing due to its small genome size. This is very crucial, as because if the genome is small and it contains less of a junk DNA, then it is easier to study such a system and to understand any changes which are being happening due to some treatment or not. So the small size, its rapid life cycle and its ability to grow without specialized growth conditions has really made it a model plant. It also helps in understanding the evolutionary aspects of biology. Next member in the monocot community is Lemna gibba. It belongs to the family Araceae and tribe Lemna, genus Lemna and species Lemna gibba. It is supposed to be the smallest flowering plant, and its growth assays are readily available, and it can be used to study the toxicity assays, and ecotoxicological studies are being done using Lemna gibba. And recombinant expression of economic production of complex biopharmaceuticals can be easily done using Lemna gibba. And demonstration of population growth curves are also done in Lemna gibba. Now we are coming to the angiosperms. In this, angiosperms can be divided into terrestrial monocots and aquatic monocots. To start with is terrestrial monocots. The first name that strikes our mind is maize, and it belongs to the order again Poales, family Poaceae, and genus G, and species is Maize. And it is suitable for microscopic studies. It is very friendly to study under microscope, and for the first time the discovery of transposons or the jumping genes was being done by Barbara McClintock using maize only. So this is a marvelous discovery in the field of molecular biology, wherein we could understand the concept of jumping genes for the very first time in maize. Next we are coming to angiosperms and aquatic dicot. In this community we have got Victoria amazonica. It belongs to the order Nymphaceales, family Nymphaceae, genus Victoria, and the species is Victoria amazonica. It is a very nice system to study. The flower is basically a white in color, and it blooms in night, and the color of the lily changes, as you can see here clearly, when it is pollinated to a pinky purple color. So you can very well distinguish between a flower which is unpollinated and a flower which is already pollinated one by change of color. This lily is being pollinated with the help of beetles, and what this flower does, that when it wishes to undergo pollination, it starts secreting some smell in terms of butterscotch smell, and this butterscotch smell actually invites the beetles to pollinate this Victoria amazonica, and moreover it provides some extra heat, which is again a lucrative offer for the beetles. And we can see that the amazing features are, it can hold up to a weight of 136 kilograms. Can you imagine the strength of the flower? And it has got a 46 centimeters of diameter. And what else? See the beauty of this life form, that it grows and nurtures some thorny features and at below their leaves to prevent or to protect itself against the predators. Now we are coming to another member, that is angiosperm, and in this community it can be again divided into terrestrial dicots, and we have got the member of model plant is Arabidopsis thaliana. It belongs to the order Brassicales and family Brassicaceae, and the genus is Arabidopsis, and finally it is called as Arabidopsis thaliana. Its small nature, short generation time is making it the most suited model plant, and it is supposed to be the plant whose genome got sequenced for the first time, and it is capable of production of several seeds, which is supposed to be one of the ideal properties of an ideal model plant. It can be used to carry on floral dip experiments, and light microscopic studies can also be done very easily using Arabidopsis thaliana. It is important plant for studying the science including genetics, evolution, population genetics and plant development, and understanding the genetic, molecular and cellular biology of flowering plants is most suited with Arabidopsis thaliana. Next member of this group is Lotus Japonicus. It belongs to the family Fabaceae and genus Lotus and species Lotus Japonicus. It is a model to understand the symbiotic nitrogen fixation, another remarkable discovery in the field of plant sciences. Its small size and generation time and short generation time has made it a suited member as a model plant. And most of the mutagenesis experiments are being done using Lotus Japonicus. Next member is Medicago truncatula. It belongs to the order Fabales, family Fabaceae and genus Medicago and species Medicago truncatula. It has got diploid genetics, its small size and easier methods of transformation, short life cycle and has got high level of natural diversity that makes it a suited member of as a model plant. It is also suitable member to understand symbiotic nitrogen fixation. It is most important member to study legume biology. Now we are coming to Mimulus guttatus. It belongs to the order Lemellus, genus Mimulus and the species is Mimulus guttatus. It is mainly used to study the evolutionary and functional genome studies. It is a model system for studying the genetics and genome basis adaptation and speciation studies. Now we are coming to Nicotiana benthamiana. It belongs to the order Solanales and family Solanaceae and genus Nicotiana and species Nicotiana benthamania. It is a model to understand plant pathogen study, and this is supposed to be the most vital place wherein the researchers are interested tremendously to understand plant pathogen interaction. Leaves can be injured easily to understand ethylene synthesis. Means any kind of stress leads to generation of ethylene. This type of studies can be easily done using Nicotiana benthamania. It is an excellent target for agroinfiltration and platform for industrial synthesis of recombinant proteins, because there are many proteins which can be overexpressed only inside the plant system, and under such circumstances we are really depending on such a plant wherein overexpression is really very facilitated. And in this community we have another member called Nicotiana Tobecome, which helps us to understand the plant physiological study at the cellular level. It has got high homogeneity and growth rate, serves as an important plant model characteristics. It helps us in understanding cell division, cytoskeleton studies and signaling pathways. Role of hormones on different plants are being studied. It helps us in cellular trafficking and it helps us to study the organelle differentiation as well. Now we are coming to populus. Populus belongs to the order Malpighiales, family Salicaceae and genus is Populus. It is a model in forest genetics and woody plant studies, and its small size and it's easy to transform. That's why it is considered as a model plant. It helps us in study wood formation and its long-term perennial growth and seasonality are the obvious areas of research interest. It is a flowering plant, understand flowering, then biotic interactions and evolutions of adaptive traits. Reproductive traits are used to study and analysis of natural genetic phenotypic variations are being done using populus. So, in today's lecture on plants as model organisms, we have initially classified the plants into their different classes, and from each class we have taken one or the other model plant, and that has really helped us to study the plant pathogen interactions, the importance of plants as serving as model organisms for overexpression of some proteins, to understand various stress related factors which are imposed on plants, and to study the effect of various xenobiotics. And this study has not only helped us to understand cell cycle regulation, it has also helped us to understand the various effects of various metabolic compounds on plants. And these informations are really helping us to understand them in a better way, so that we can use them for the betterment of mankind. So this is what we have discussed in our today's lecture.